Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Good afternoon. My name is alex, and today I have the pleasure to introduce SeungGÇÖs, who's here to join us as part of the Microsoft Research Visiting Speaker Series. Sebastian is Professor of Computational Neuroscience in the Department of Brain and Cognitive Sciences and the Department of Physics at MIT, as well as an investigator at the Howard Hughes Medical Institute. He's been a Packard Fellow, Sloan Fellow, McKnight Scholar, and PopTech Science Fellow, among many honors. And the last time I was so excited to introduce someone was when I first heard about someone trying to map the human genome. But that's been done already. So now move on to the next thing. And Sebastian is trying to work on mapping the human connectome. And if you don't know what that is, I hope that you will by the time the Sebastian talk is done. So please welcome me in welcoming Professor Sebastian Seung. And I hope that you'll be interested in this afterwards.
Thank you very much, Alex. And thank you all for coming. As you saw, I've written a book. It's been published. There's something paradoxical about writing a book, which is that you start out with the idea, oh, I'm going to share my ideas with the world. And then you spend the next few years of your life in solitary confinement writing this book and suffering. But now that's all past. So I'm just having a really good time sharing with you. So thanks a lot for coming. This is also special. It's my first trip to Microsoft. And of course, Microsoft has been a legendary force in the history of computing, and in particular in the personal computer revolution. And I think that computers have come to have a kind of mythic significance for us, right? That, in fact, people come to MIT, I guess freshmen will come and say, you know, my dream is to build a robot that thinks, or a computer that is more intelligent than I am, right? And in fact, I just talked with Alex and his colleagues about the artificial intelligence research at Microsoft. And so this activity is still alive and well. It's been going on since, I guess, 1950, the whole field of AI. And as any kind of mythic quest goes, sometimes people think about it in an apocalyptic way. They think that maybe in the future computers will become so powerful that there will be no place for us left in the world. So there's this kind of larger-than-life significance to the whole quest to find or to construct artificial intelligence. So what I'm going to tell you about today can also be thought of as another quest, a parallel quest, one that is turned upside down. And that is the quest to deconstruct our own brains, to reverse engineer them, to figure out how they work. And the connectome really is a radical vision of how it's not going to be realized in a really short time, just like AI won't be realized in a short time. It's a multi-generational quest to understand ourselves, to know ourselves by deconstructing our own brains. Now, the connectome, I will first define what a connectome is, I guess. I will show you the only animal for which an entire connectome has been mapped. This is a worm, a 1 millimeter long worm that lives in the soil. It's called C. elegans, and it's one of the favorite organisms that's studied by biologists today. I think it's kind of cute. I don't know about you. Not so much? All right. Well, here is its connectome. So I don't know about you, but this always reminds me of the back pages of in-flight magazines. Maybe I'm spending too much time on planes these days, but imagine that every city in the airline route map is replaced by a neuron. In this case, the neurons all have names, actually. And every route between two cities is replaced by a connection between two neurons. So this little worm has 300 neurons, just 300 neurons and 7,000 connections. And it's already pretty complicated looking, right? Now let's imagine your connectome. Your connectome has the equivalent of 100 billion cities and 10,000 flights coming and going from each city. So this kind of gives you the idea of what a connectome is, but it really doesn't measure up at all to the scale of what your connectome is like. An extremely complicated map. You might ask me, is this map universal for all worms? Well, all worms, all normal worms, have the same neurons. The neurons have the same names, they occur in the same locations, they have the same shapes, and there are some small differences in the connections. So there's a little bit of variability from worm to worm in the connectome. In the case of you, your connectome and mine, the connectomes of all of us, are much more different. There's a lot more variability than a worm connectome. And maybe that's why we like to think of ourselves as more unique. Each of us is more unique than maybe a particular worm is. And there's much more variation in your connectomes than between your genomes. You may know that the differences in genomes between people are really, really minuscule, right? Only a few letters out of the entire, a small percentage of letters out of the entire sequence. Yeah, so that's a connectome, and why should we actually study connectomes? So that is something that I try to address in the book. So there's a lot of how-to in the book. How are connectomes found? How do scientists go about studying them? But more importantly than that, we have to explain why do we have to study connectomes. So there's an older tradition of studying the brain, which all of you are no doubt familiar with, which is to take the brain and divide it into regions. You've heard about left brain and right brain, and maybe you've heard about frontal lobe, parietal lobe, temporal lobe, those names, right? And there's some somewhat finer subdivisions, and neuroscientists have ascribed various functions to these regions, and you can interpret the results of symptoms of brain injury in terms of these kinds of maps, and so on. So we've learned a lot about the brain in this way. But the regional approach to understanding the brain is never going to answer the questions that we really care about, in my opinion. So what are those questions? So one is, what makes a brain region work well, let's say in the case of a genius, or badly in the case of someone with a mental disorder? What happens to a brain region when we learn something? There's a kind of primitive answer, right? People have tried to look for differences in size. The 19th century neuroscientists thought that studying was like working out, right? Maybe your brain gets bigger when you study. And surprisingly, there is some evidence that on average, parts of brains can enlarge when you're studying. But you can only see these differences by averages over large populations of people. And the same thing holds with certain mental disorders. So let's talk about disorders like, well, first let's talk about disorders like Alzheimer's. With Alzheimer's disease or Parkinson's disease, we know that neurons degenerate and die. If you look at the brains of these people at autopsy after they're dead, there's clearly something wrong with the brain. There's junk accumulating in the neurons, they're dying neurons, so on and so forth. But if you look at the brains of people who had autism or schizophrenia, in general, their neurons look healthy. People have looked hard to find brain abnormalities that are clear and consistent in the cases of these mental disorders. And again, they resort to things like crude measures like size. On average, autistic kids, 2-year-old autistic kids, have larger brains than those of typical kids. But if you took every kid with a big head and said that kid's autistic, you'd be woefully inaccurate. And that's because studying brain regions, or the size of the brain, or its regions, is just too crude. So in order to answer the questions that we really care about, why do brains work well? Why do they work badly sometimes? And what happens when we learn? We have to study the brain at a level that's smaller than regions. We have to subdivide regions and study their parts, which are neurons. And in order to figure out how regions work, it's important to figure out how those parts are organized. And that's why we're trying to find connectomes. You guys are all engineers, so you would be familiar with the idea that an electronic device, in order to understand how it works, we might want to find the wiring diagram. So that's another way of thinking about it. Okay, so why are connectomes important? Well, more specifically, the second part of the book makes two claims. One of them is that connectomes are not static, but they change. They change they change as the result of experiences. So when you have an experience, and you now store a memory of that experience, there's some indirect evidence that your connectome gets rearranged. The connections change in some way. And that's how you store a memory of an experience. The second reason that connectomes are important is to understand the relationship between genes and the mind. Genetic factors are also important. So we all know, right, that usually people are kind of like their parents. And in fact, one of the important stages of life is when you come to the realization, I can't believe I'm behaving like my parents. It happens, right? So is that the result of your upbringing? Is that the result of your genes? You get both from your parents, right? And people have had holy wars about this. They get really mad at each other. They call each other names, maybe even fist fights, right? But we have to move beyond that, because I think everyone now realizes, at least all scientists agree, that of course both genes and experiences are important. They both shape the mind. To move beyond that, we have to understand exactly how do they shape the mind. How do experiences change the brain? Well, maybe they change the connectome. How do genes shape the brain? There's a number of ways in which they do so, but one important way is by guiding the process by which the connectome forms during development. So as a brain grows, many steps have to happen. 100 billion neurons have to be born. Then they migrate to their proper positions inside the brain in a complicated dance. When they get there, they send out branches, these long branches. You've seen pictures of neurons before. They have these incredibly beautiful branches. The branches intertwine with each other. And at the points where they contact, they can make little junctions called synapses. Synapses are where two neurons talk to each other. So that's how a connectome gets formed when you're growing up, and genes are important for all of those processes. So that's an important way that genes influence the brain. So I would argue that the connectome is where nature meets nurture. That's the meeting point. And if we want to understand our own personal identities, we have to understand connectomes. If we want to understand how to change ourselves, we need to understand connectomes. So you can argue that every important transformation in your life is about changing your brain, right? Going to school and learning something, falling in love, being a more compassionate spouse, healing from a brain injury. All of these things are changes in your brain. And I'm arguing in the book that they're fundamentally about changing this map. So that's the motivation for looking for connectomes. How do we go about finding them? Well, I should first say that this map took over a dozen years to find. It was a heroic effort in the 1970s and 80s. So given that took a dozen years, then how could we even hope to find connectomes of human brains, or let alone let's say mouse brains. Mouse brains would be smaller, right? How can we hope to find connectomes of brains that are more like our own, not as primitive as that of this worm? So the hope is that our technologies have advanced far beyond what we had in the 70s and 80s. A lot of it is advances in computers. And in fact, the hope is that computers are what are going to enable us to study the brain. So I made this joke earlier as I was talking with Alex and his colleagues, which is that AI researchers have long look to the brain for inspiration to make computers smarter. Right? If we copy the brain, maybe we can make computers smarter. But we've reached a new stage where computers have become very smart. They've improved a lot in the last decades. How can we use computers to help us figure out how the brain works? That's what my lab is all about right now. So my joke is, ask not what the brain can do for the computer. Ask what the computer can do for the brain. So I'm going to show you the process by which this is done and the technologies that are being developed in order to speed up this process. One important step is imaging the brain. So you take a piece of brain and you put it in a high-tech deli slicer fitted with the world's sharpest knife, a diamond knife. And you cut it into slices that are 1,000 times thinner than a hair. And you array all the slices on a silicon platter and you put them in a scanning electron microscope. And I'll show you images that were taken in the laboratory of Jeff Lichtman at Harvard University, who collaborated with researchers at Microsoft and got some funding from Microsoft in the early stages. So we're going to zoom in on one slice of a brain by a factor of 100,000 times. And you see round circles right now. Those are cell bodies. They're the round boring part of the neuron that has the DNA and the nucleus. And then we've zoomed into the spaces between the cell bodies. Those are the entangled branches of neurons and synapses, although you may not recognize that yet. Now, if we take many images of slices and stack them up together, we get a three-dimensional image. And still, you may not really see anything that looks like a neuron. So what we'll do is color in the cross-sections of one branch of a neuron through the slices. Slice after slice. And if we do that, we get a three-dimensional reconstruction of what this little segment of a branch looks like. The red is a branch called a dendrite, and the green is a branch called an axon. And the green touches the red at two locations. Those are the synapses. And we're zooming in on one synapse. And if you look at the interior of the green neuron, you'll see little circles. So those circles are vesicles. They're little bags containing molecules called neurotransmitters. You've probably heard of neurotransmitters before, right? Serotonin, dopamine, glutamate, these things. They were abstractions when you heard about them. Now you can actually see where neurotransmitters are kept inside neurons. So this green neuron has sequestered the neurotransmitter molecules, and when it wants to send a message to the red neuron, it expels the contents of one of those vesicles into the cleft between the two neurons. Then on the other side, that red neuron has receptor molecules which sense the neurotransmitter molecules and thereby receive the message. So that's an account of what's called synaptic transmission. That's how neurons communicate, and you can read about that in textbooks. But to drive the point home, let me ask you to reflect on what this means. So every time that you are thinking or feeling even the most refined ethereal thoughts, effectively your neurons are spitting on each other. Right? That thinking is an untold number of microscopic secretions. And in fact, there was a French philosopher who wrote a long time ago, centuries ago, that as the liver secretes bile, the brain secretes thoughts. And amazingly, he turned out to be right. Okay, so chemical signals transfer messages between neurons. Electrical signals are traveling inside neurons along the branches. That's kind of the basic means of communication inside the brain. So you've seen how to find a synapse. Now, how do we find a connectome? Well, if you take that entire cube, imagine coloring it in like a three-dimensional coloring book, which is quite a laborious task because there's lots of objects besides those two I just showed you. I showed you those, but in fact, that whole volume is packed by branches of neurons. And the embarrassing thing is that this looks like a fancy computer-generated animation, which it is, but it was created by a person, Daniel Berger, a postdoc in my lab, who painstakingly colored in, slice by slice, every branch in there, and it took him about 250 hours of work. And to give you an idea of the scale, that's really pretty small compared to an entire neuron. That has only small pieces of branches. And that neuron is pretty small compared to an entire brain. And that's just a mouse brain. So as you can see, we're very far from finding the connectome of an entire human brain, or even a mouse brain. We'd be happy with maybe a cubic millimeter piece of mouse brain or human brain. That would be a lot of work. If Daniel were to color that in, it would take him somewhere between 100,000 and 1,000,000 years if he never took a nap. Right? And this is a problem that's people are encountering throughout our society. Our ability to generate data is far outstripping our ability to analyze it. At Harvard, they'll be taking delivery from Zeiss of a very fancy new microscope. It has 61 beams that scan in parallel, and it will image a cubic millimeter of brain every 2 weeks, generating 1 petabyte of data. That's going to arrive, I think, in a year and a half. There's no way Daniel can keep up, right? So what can we do? Well, there's 2 things. One is the obvious one of artificial intelligence. Let's make computers that are smart enough to do what Daniel does. Computers that can see. My lab has worked on that, and other labs around, there's a few other labs around the world have worked hard on that. We've made some progress. We use the machine learning approach. That's a little bit new in this area. It's a problem known as image segmentation. And so we use Daniel's coloring as the training set. So we trained the computer to emulate Daniel on that cube. And we've gotten good results, but still the error rate is still high. It's not anywhere nearly as accurate as we'd like it to be. So that's a problem. You know, it's natural, right? We know that robots in real life don't see as well as they do in the movies. That's just the way it is right now. AI is not perfect. So what we have then is we developed human-computer interface that allows a human user to interact with the AI and fix the errors. And that semi-automated system can be up to 100 times faster than Daniel coloring all by himself. But that's still not enough, right? I told you 100,000 person years to get a cubic millimeter. So we have to make the AI smarter. And the other thing we can do is we can try to recruit more people. So here's this new website that we've launched. It's called iWire, EYE-wire.org. A bunch of students at MIT actually helped us launch this. We have a special January term, a 4-week term that's between the semesters when the wild and crazy classes are offered. And so I recruited a bunch of undergrads to help us launch this website. So you can go there and you see this invitation, welcome to iWire, where you can help make discoveries about the neural structure of the retina. So the retina is the sheet of neural tissue at the back of the eye, and we want to find the connectome of the retina. We want people to help us discover it, right? So the great thing about this site is you can come and you can click on these various tabs. You can learn about neuroscience, and then you can proceed to the next tab and actually do some neuroscience. You can contribute to what's known about the retina. So it's a game, and the game is something like what you used to do as a kid with a coloring book. I hope this will appear. The connection's a bit slow, I guess. There it is. OK. So on the right-hand side, what you see is one slice through a little cube. Right? So we take the entire image that we have, which is a very large image. We divide it up into little cubes. Those cubes are roughly of the same size that you just saw. And we can display one slice at a time on the web browser. On the left-hand side here, you see a 3D rendering. Here's the cube. And the yellow right here indicates which slice of the cube we're looking at on the right. And the AI has colored in one branch of a neuron starting at that point. Its 3D shape is shown right here. It's rendered like this. May still be loading this image. Let's see. All right, I'm going to have to keep on talking because I think this image is loading very slowly, unfortunately. I guess it's there. Oh, there's one thing. I apologize that I'm showing this in Chrome, and we don't support Internet Explorer. And there's a reason, which is that we don't have WebGL support. So I want to talk to somebody in Internet Explorer who I can persuade to support WebGL so that we can make this run in IE. Yeah, unfortunately, the demo is not working too well here because it's taking too much time to download those images. Yeah, so this works well on a right now a 10 megabit per second connection, but you can see what the AI has colored in, and try to move down. And find a place where the computer has failed to color. So you can see right here, the AI has failed to continue coloring. So right here, the AI should have kept on coloring. And we can try to correct that by simply clicking. OK? So we just tell the AI keep on going, and it colors in there. And it should be coloring in the adjacent slices, too. All right, I'll come back to that. I'll run it after the cube is cached. All right, so the basic idea then is that, and I should say this is a tutorial task. So here, an expert has already done this task. So the right answer is known. And the new user does this and gets feedback. If the user clicks in the wrong location and colors in the wrong object, then that will show up in red. Well, maybe not. All right. Well, I'll let this cube cache. So you get the person in the training gets negative feedback if they color in the wrong thing, and they also get a progress bar right here, which tells them how far they've gotten. But eventually, once they get good enough at this, they will get cubes that no expert has done before. And then they can actually help to reconstruct, trace the wires of the retina, and help us discover how the neurons are connected. OK, so we've gotten a lot of enthusiastic response from this, especially from educators, too. So high school students are going to be able to learn about neuroscience and do neuroscience at the same time. So we think this will be a very powerful model. We have also been exploring partnerships with patient advocacy groups, because this could be a powerful way of mobilizing people to study brain disorders. And which brain disorders could we hope to do? Well, I told you that in the cases of autism and schizophrenia, clear and consistent neuropathologies have not been found. One really intriguing hypothesis is that the brains are miswired, that the neurons are all healthy, but they're connected in ways that are abnormal. And we've never had the technology to see such miswirings before, but we are planning to image brains and compare regular brains with autistic or schizophrenic brains and see whether we can find differences in the wiring between those brains. So those are called, there's a new jargon which we say is connectopathy. A connectopathy is a pathological pattern of connectivity that's associated with a mental disorder. So that's going to be a very powerful model, and I think that finding a connectopathy would be a huge advance in our understanding of mental disorders. Because if you can't see something wrong, I think it's much more difficult to find a treatment or a cure for it. In the case of infectious diseases, imagine not having a microscope and not being able to see bacteria. That's a problem, right? If you don't see the bacterial cause of an infectious disease, all you see are the symptoms. But once you see what's wrong, that doesn't cure it by itself, but that helps you find a cure because you know what you're trying to get rid of. And in the same way, I believe that finding a physical basis for mental disorders in the brain is extremely important. Is it working? A little better. All right, well, too bad about that demo. Okay, so I've shown you the technologies that we're using to find connectomes. It's including artificial intelligence and now using the mechanisms of social computing to bring people together to collectively work with artificial intelligence. And the beauty of that, of course, is that when we take human judgments, we can use that for machine learning to make the AI smarter, progressively smarter. So we're trying to create a system that collects the human examples of these tasks, and it's going to get more and more powerful over time. Now, I've portrayed the study of connectomes as really the way that we want to come to grips with the notion of change, personal change, and really arrive at a science of personal change that is a rigorous and real one, not like the one that you find in self-help books. Right? Self-help books usually these days they all have a lot of neuroscience talk, but the reality is that we don't know what happens in the brain when we change. The last part of my book discusses the most extreme dreams of change, which are those that belong to the transhumanists. So you may have heard of their two biggest dreams, which are cryonics and uploading. So cryonics is the practice of freezing your brain after you die in the hope that some advanced future civilization will resurrect you. And uploading is even more extreme. It says, let's get rid of our bodies altogether. Let's just transfer our minds to computers. Let's simulate our brains in computers, and then we can live forever inside this electronic heaven. Now, these are science fiction dreams, and for the most part, dialogue about them is either, I believe this and I'm signing up for it, or these people are loony. And there's not really any kind of rational debate. So in this book, I argue that if we use the notion of the connectome, we can interestingly subject some of these claims to scientific tests. How is that? Well, if you believe that you are your connectome, that your memories are encoded in your connectome, then the critical question for cryonics is whether their freezing procedure preserves the connectome. We know that brains start to disintegrate, deteriorate as soon as you die. Are the wires falling apart? Are the synapses falling apart? If that's the case, then our advanced future civilization might be able to resurrect your body, but it couldn't recover your memories. It wouldn't be able to resurrect your mind. And so that's the critical question for cryonics. We have no idea. They say their procedure is going to preserve brains, but the really critical test is to see if it's preserving the connectomes in those brains. And I think that's science because we have the potential of falsifying the claims of cryonics. Anytime you can falsify, a potential for falsifying something, you're starting to enter the domain of science rather than philosophy. Philosophy, you can never prove anything wrong. And in science, it's hard to prove anything right, but you can prove that things are wrong. Now, what about uploading? Why is the connectome important for that? Well, the only remotely plausible proposal I've heard about uploading is based on the connectome. We take you, let's say you're my customer for uploading, you give me your brain, we find your connectome, and we use that information to simulate the network of neurons inside your brain. And in fact, you could imagine that if we're optimistic, if Moore's law continues and computer technology improves at the same rate it's improved for the last 50 years, we can imagine that by the end of this century, we could have computers powerful enough to both find human connectomes and to simulate an entire human brain's neural network. So the crucial question is, are you really your connectome? Is that really all the information that's needed? Or could there be other information in your brain that's not in the connectome that is critical for your personal identity? And so I discussed those issues. I discussed the possible pitfalls, what extra information might be needed in order to create one of these simulations. And so the problem is that if a lot of extra information is needed, then those computer simulations become completely impractical. There's no way we would ever do them even after 100 more years of computer progress. Okay, so that's the summary of the book. I know that books, you read books partially because of ideas, but you also read them for fun. So I think I will just give you a flavor of the writing. I'll read a little bit from the book. Can't decide which chapter to read. All right, I'll read the chapter that discusses the imaging technology. It's called Seeing is Believing. Smelling whets the appetite, and listening saves relationships, but seeing is believing. More than any other sense, we trust our eyes to tell us what is real. Is this just a biological accident, the result of the particular way in which our sense organs and brains happen to evolve? If our dogs could share their thoughts by more than a bark or a wag of the tail, would they tell us that smelling is believing? As a bat dines on an insect captured in the darkness of night by following the echoes of ultrasonic chirps, does it pause to think that hearing is believing? Or perhaps our preference for vision is more fundamental than biology, based instead on the laws of physics. The straight lines of light rays, bent in an orderly fashion by a lens, preserve spatial relationships between the parts of an object. And images contain so much information that, until the development of computers, they could not easily be manipulated to create forgeries. Whatever the reason, seeing has always been central to our beliefs. In the lives of many Christian saints, visions of God, apocalyptic or serene, often triggered the conversion of pagans into believers. Unlike religion, science is supposed to employ a method based on the formulation and empirical testing of hypotheses. But science, too, can be propelled by visual revelations, the sudden and simple sight of something amazing. Sometimes, science is just seeing. So why don't I take a few questions now? Oh, the name of the book is just Connectome. Connectome. Yes, question?
You have the connectome of C. elegans. What have we learned from it?
So if you talk to C. elegans researchers, in fact, everything we know about the C. elegans nervous system, the functioning of it, really depends on this diagram. On the other hand, you could argue that we've learned disappointingly little about how it works, right? So another way of asking this is, what's missing? And so what I would say is missing is that there's almost no measurements of neural signals inside C. elegans. Because of technical reasons, it's easier to measure neural signals inside the brain of a mouse or a cat or even a human, the signals of a single neuron, than it is to measure them in the worm. So the worm was actually an unfortunate choice, it turned out to be. And so you need both. You need both signals, the neural activity, and you need the connections.
So you have the connectome, and you don't know why, what genes is it used for its mobility or sensing or eating or anything?
Oh no, a lot of those things are known because you know that those neurons are many of them are classified as sensory neurons, and we know which sense they touch or smell or whatever they correspond to. There's motor neurons, and we know which muscles they actually control. And a lot of reflexes, stimulus response behaviors have been worked out because you can find a pathway from the sensory neuron to the motor neuron. But at the same time, there's a lot of things that haven't been completely understood. And the big stumbling block has been the lack of measurements of activity. And you need both. So there's three things you have to understand, three kinds of quantities you have to understand in neuroscience. That's genes, connections, and activity.
In terms of Moore's law extrapolation, you might have the capacity to understand a connectome at some point because you've got all that processing power available. But you have to be able to ask the questions and set the model up and so on and so forth. How's that going to work?
Oh, well, so I'm just predicting that. So the model would be built by finding a connectome, right? Taking an entire human brain and slicing it up and imaging it at really high resolution and extracting the connectome. And I'm extrapolating that I'm assuming that that's primarily a computationally limited task.
Right. Okay.
Yeah. That's right. So both the simulation and the finding of the connectome are going to ride Moore's law, or if Moore's law falters, those efforts will falter too.
Yeah, so comparing connectome versus genome, right? So once you get the genome out, you still have to do a lot of work to find the genes. From that, you need to infer the functions. Now, given that your connectome is found already, what else do you want to do?
Wait.
Do you want to look at more functional?
So you're asking, if I had a connectome, what would I do with it?
Yeah.
All right. So as long as we're engaged in wishful thinking. So I say that there's, I actually have 4 chapters in the book for that. I have carving, code breaking, comparing, and changing. So carving is kind of like, you take the genome and you divide it up into genes, right? So carving a connectome would be to divide up this graph into brain regions and groups of neurons that are called cell types. So that's one thing we would do. Code breaking is to try to read memories from connectomes. If memories are really encoded in these patterns of connections, we have to figure out how to decode them. And comparing, that's the obvious one, right? So healthy brain with mental disorder. We have to compare those two. And then the final one is to have better technologies for changing brains.
In addition to connectome, there is also chemistry, proteins. Maybe too much depends on them. Even if we know all connectome, it's possible that we just structural equal.
Yeah. So remember, I said genes. So the question. This really goes back to genes. This triad of genes, connections, and activity, right? So genes are a shorthand for the detailed identity of molecules that are found inside neurons. And so in order to model or understand how a network functions, I might need to know exactly what kinds of molecules are at each synapse, exactly what kind of molecules are distributed throughout the dendrite of the neuron, so on and so forth. So you're adding to that, or are you? Okay, let me just finish that. So you're right, but hopefully here we can use the intermediate step of cell types. So neurons, you can think about them as trees in a forest. They fall into different species. And ideally, neurons of the same species, which anatomists call cell type, they will have similar molecular properties. And there'll be a finite number of them, a relatively small number of them. So if we just characterize the molecules that are in each type of neuron, that may be enough. If not, then one might have to have more advanced imaging technologies that would really see not just a grayscale image like this, but really figure out exactly what molecules are where. And if all that information is really necessary, that's when the uploading people, I think, they have to give up. Right? Then it becomes really complicated to simulate. Yeah.
I was wondering if you've seen any evidence so far that as you're trying to build these models of the connectomes, that there are fractal-like repeating patterns, or is like everything not like anything else?
Are there repeating patterns? Well, we haven't seen enough of the. Well, okay. So let's just. There's obviously repeating patterns in certain structures like the retina. Right. So the retina is a sheet of tissue, and one part of it over here looks very similar to another part over here. Now, on the connectional level, you can see repeating patterns, but they're too small to be really that interesting. Let's say a group of 3 neurons that have a certain pattern of connection between them. That kind of repeating pattern has been observed, but
Too micro, too local.
Maybe it's too local to be really that interesting. So the challenge here is to find larger patterns that repeat.
If you have multiple neurotransmitters in a synapse, is the information passed like a byte where the amplitude of the neurotransmitters in that synapse is the signal of each?
So you're asking about whether a synapse has some kind of analog strength? Is that the question?
Well, yeah, whether the transfer across that, depending on the information it's transferring, has analog strength in the synapse.
Yes, so a synapse, when a neuron signals another neuron through a chemical synapse,
Right.
the effect of that on the receiving neuron has it's an electrical effect in the end, and that electrical effect has an amplitude. And that's often called the strength of the synapse.
But there's more than one neurotransmitter type. Do
Usually,
they transfer different information, or are they all transferring the same information?
well, they're transferring different kinds of information. So, for example, most excitatory synapses, the positive synapses in the cerebral cortex, are glutamate synapses. And the ones that are inhibitory, synapses with negative sign, are GABA. That's another molecule that's similar to glutamate. So yes, different neurotransmitters have different effects. But a single neuron typically secretes only one neurotransmitter.
That's what it is.
Yeah. One in the back?
So from online, is there any technology that would allow us to get our connectome without slicing our brain?
So you may have heard of the NIH-funded Human Connectome project, which uses MRI, is using MRI to find the so-called human connectome, but that's a very different scale than what I'm talking about right here. So they're trying to find the map of connections between brain regions, and they can do that with non-invasive imaging in live brains. But that's really simple compared to the neuronal connectome. We don't know of any technology that could do that in a live brain. Or we can maybe imagine some technologies that might do that in a live brain, but maybe it wouldn't make any sense because it would take so long. It would take longer than your lifetime to achieve it. So the connectome would be changing during that whole time when you're imaging it. But the basic answer is no. We don't know of any. On the other hand, we don't know of any law of physics that tells us it's impossible. So I can't rule it out that someone will invent one in the future. But right now, we take dead brains and we slice them up. Too bad. Yeah.
Could you use, for instance, could you use MRIs from live people to inform how you would go looking for the connectome or related connectome issues? For instance, if you had abnormal MRI results, is it feasible to say, like in a mouse that you did stuff to, if you screwed with a mouse on purpose, did MRI stuff on the mouse, and then you could use...
Well, MRI is useful for telling us where to look.
Yes.
So MRI will tell us, look at this location in the brain. And with this high-resolution method, we can only do little pieces anyway. So we need a lot of guidance about where to look. That's one way in which these two... I mean, that's typical of maps, right? You've got zoomed-out views and zoomed-in views. And you use the zoomed-out view to tell you where to look.
Does your model include the strength of the synapses? And if so, can the imaging determine that?
So there's a lot of evidence that the strength of the synapses is well correlated with their size. But that relationship remains, it needs more investigation. And so it may be, and more than that, it may be that the strength of a synapse, it doesn't even make sense to summarize it by a single number. Synapses are more complicated than that. They have more complicated dynamics. So we may need imaging methods that have more sort of molecular resolution, like this gentleman asked about before, in order to really try to extract all the functional properties of a synapse from its appearance. Remains to be seen. And so the strength of a synapse, just to fill this in, so a synapse has, a connection has an efficacy at transmitting information, which is analog. And so some people believe that memories are stored by changes in that strength. Other people argue that memory storage also involves creation and elimination of synapses, not just changes in the strength of the existing ones.
Is the structure of connectome fully encoded in the genome?
No, it's not.
So to build an original...
Because experiences also...
No, experience is on top of it, but original structure of it doesn't go from genome. It goes also from something else, right?
Well, you could think about the original, the starting point. Experience starts working on a structure that is created by genes and randomness. Right, so there's really not just genes and experiences, there's also genes and randomness. But probably, it's not like there's one stage in your life where genes are doing all the work and then all of a sudden it switches over to experiences. I'm not sure there's any way of separating those two into separate phases of life. Genes and experiences are always working together. Even in the womb, there's still some kind of experience taking place. Yeah.
You mentioned that you're using machine learning to automate the coloring process or nerve tracing. Could you comment on other algorithms that you may be using in their application area, or is that the primary one that you're up to?
I mean,
therefore, are you or colleagues in your adjacent office next to you in your center working on algorithms for understanding the structures as the structures are coming up? Say unsupervised learning and applying them somewhere.
Well, so you're asking about whether we're doing things that are not learning based, or whether we're using supervised versus unsupervised learning?
It's a more broad question. Besides the one case you gave, what would be another example? Are they illustrated in the book?
I don't say much about it in the book. You'll have to go to our review articles. So the major thing that we've done in image segmentation has been supervised learning algorithms that are based on true metrics of segmentation performance. So the classic, I guess not even classic, but the 10-year-old machine learning approach for image segmentation focuses on deciding whether a pixel is either a boundary or not a boundary. But the problem with that is that pixel error is a very naive error. If a human tracer traces the boundaries of an object, there's a lot of jitter in the placement of the human judgments that we really don't care about. What we care about are differences that cause topological consequences, like merging two objects or breaking an object into two. And so we have a machine learning framework in which that's what's penalized, as opposed to small jitter in boundaries. That's just one thing that's needed. The challenge right now is really moving to larger and larger context. So the general problem in computer vision always is that your judgment about what happens right here can depend on things that are happening really far away. And so how do you integrate this contextual information into local decisions? We have one question here.
Can you say something about the relationship of connectome to other effort in trying to understand how the brain works? Is this the only approach, or there may be some other approach?
Well, I said there's one way of thinking about it is activity, genes, connections, and activity. Neuroscientists generally study one of those three things. And another approach we can talk about is observation versus manipulation. So the connectome is just an observation. We don't actually, we're not manipulating connections. We're just observing what they are. But there's optogenetics, which involves manipulating activity by light. And one can imagine a whole other class of techniques that would involve manipulating connections, destroying a class of connections to see what consequence that has for function. Yep.
So Jeff asks, can we map a small volume of brain and understand anything about what behavior that cube is responsible for?
Well, what we would do is, ideally, we would measure activity in that cube in a behaving animal. So then the activity is related to behavior. And then we take that cube out and then find its connections. So that's sort of a fourth, right? So there's genes, connections, activity, and behavior. And we can apply them all to that one cube.
One more question.
So is there any notion that there's some computational codes such that there's something called the software in the brain when you get to a higher level?
You mean a hardware-software distinction?
Yeah.
Well, at a high enough level, yeah. So if you were asked to simulate a compiler, then you would probably you'd be working at sort of a software level, right? But there's a lot of reasons to believe that the brain is not like a general-purpose computer. One of them being that particular brain regions are specialized for particular functions. Right? So in a universal computer, we have one CPU which does all functions. But in fact, our brains have different regions which are specialized to separate functions.
So wouldn't that be equivalent to like a graphics chip? And such that at some point you have to get up to the level where these things can be encoded?
Yeah. Yeah. So at the level of the things that we don't do very well, like abstract thought, those might be more like a software thing. But most of the functioning of the brain probably isn't like that.
Lots of fixed function.
What's that?
In the heterogeneous computing world, we call that fixed function.
Fixed function.
Inside a GPU, you've got some programmed and some fixed functions. Heartbeat is probably not programmed. It's probably fixed.
All right, let me. I'll just read one last excerpt, if you guys don't mind. Do we have a little time? So this is the beginning of the book. And I'll give you an image to go away with, a mental image. No road, no trail can penetrate this forest. The long and delicate branches of its trees lie everywhere, choking space with their exuberant growth. No sunbeam can fly a path tortuous enough to navigate the narrow spaces between these entangled branches. All the trees of this dark forest grew from 100 billion seeds planted together. And all in one day, every tree is destined to die. This forest is majestic, but also comic and even tragic. It is all of these things. Indeed, sometimes I think it is everything. Every novel and every symphony, every cruel murder and every act of mercy, every love affair and every quarrel, every joke, and every sorrow. All these things come from the forest. You may be surprised to hear that it fits in a container less than 1 foot in diameter, and that there are 7 billion on this earth. You happen to be the caretaker of one, the forest that lives inside your skull. The trees of which I speak are those special cells called neurons. The mission of neuroscience is to explore their enchanted branches, to tame the jungle of the mind. Thank you all for coming.